Welcome back, everybody. We're now doing session 3 of the cell biology module. The chapter that you need to have read before you've come to this is chapter 3, and we are going to learn today about cell communication. In preparation for this session, you needed to have read that chapter 3, and also it's a good idea to revise the plasma membrane structure and function, okay, so that you can understand vesicular transport a bit easier. So just a bit of recapping here, or revision. In the last session you learned about the structure of the plasma membrane. If you remember, it's made up of a lipid bilayer, and in that lipid bilayer you have phospholipids, which have a polar head and a non-polar tail. You have cholesterol, which strengthens the actual plasma membrane, and then you have your glycolipids. As well as the lipids that are in the plasma membrane, you also have membrane proteins. So if you remember, you've got two different types of membrane proteins. You've got your integral membrane proteins. Most of them are called transmembrane, meaning they go all the way through from one side to the other. And you also got, what are the other ones, if you remember? What are the other ones called? They are peripheral proteins. So those ones are either on the outside or the inside of the cell, but they don't go the whole way through. Now, all of those proteins, those proteins have 6 functions, or they can have 6 functions. So those functions, if you remember, if you're recapping, they're ion channel, carrier, receptor, enzyme, cell identity marker, and linker. What we then did, we went over membrane transport. So how things got across the membrane. Things have to come in and things have to go out. So the first one we did was passive transport. And if you remember, with passive transport, no energy is required, and it always goes from high to low concentration, and it uses its own kinetic energy. So its own energy that it creates from moving around. 3 types of passive transport. You have simple diffusion, where it goes straight through the membrane, doesn't require any help, it can just go straight through that lipid bilayer. You then have your facilitated diffusion, and there's 2 types of facilitated diffusion. You have channel-mediated and carrier-mediated. So channel-mediated uses proteins that act as channels, and carrier-mediated uses proteins that act as carriers. Do you remember what the difference between a carrier and an ion channel? Carrier channels change shape to bring the molecule in. And then the last one is osmosis, and osmosis is the movement of solvent or water. We then learnt about active transport. 2 types of active transport: primary active transport, secondary active transport. So in primary active transport, substances or molecules are moved against their concentration gradient into the cell, okay, utilizing energy in the form of ATP. So it breaks down ATP, which provides the energy to bring substances against their concentration gradient, either into or out of the cell. That's primary. Secondary active transport, there it utilizes the energy stored in the concentration gradient. And if you remember, 2 types, antiporters and symporters. So they're carrier proteins, they're proteins acting as carriers, and it either moves 2 substances in the same direction, which is SYM, same, or in the opposite direction, which is antiporter. Now, one of those substances is either, well, in the cases that we showed you, was sodium. So, okay, sodium is one of the molecules that moves back into the cell, and then we had substances that either move out or in, depending on whether it was an antiporter or a symporter. So that now brings us to where we are now, the 3rd type of transport across the plasma membrane, which is vesicular transport. So, communication. What does vesicular transport have to do with communication? Cells must communicate with each other, the ones next to each other, the ones further away, and also it has to communicate within the cell. So organelles need to communicate. So the cell itself, within itself, has to communicate so that it knows what to do. So the 2 types of communication that we're looking at is transport, otherwise known as trafficking, and that's done through vesicular transport, and signaling. Now, signaling we'll do next session. Okay, so Transport or trafficking. It is the physical movement of proteins and lipids within the cell. So it's how things move into and out of the cell and how they move around once they're in the cell. Signaling, on the other hand, is transport of a specific signal. It's received from the outside and how that elicits a response on the inside of the cell, how it makes a cell do something. Now, the two types work together. Okay, they're definitely not separate from each other. They each need each other so that they can communicate. So trafficking influences signaling and signaling influences trafficking. There are three types of vesicular transport. Okay, you have endocytosis, exocytosis, and transcytosis. They're the three types. So let's look at the words so we can try and understand the words. First one, endocytosis. So the prefix endo means into, so coming into something. So endo, coming in, cytosis, the word cyto means cell. So it's bringing things, it's the process of bringing things into the cell. Exocytosis, on the other hand, exo means external, moving things out of the cell. Okay, so exo, moving out, cytosis, cell. Transcytosis, trans meaning both. It has both endocytosis and exocytosis. So bringing things into the cell, and then they move straight out to the other side and come out the other side, which is exocytosis. If you have a look at the diagrams over here, this one here, you've got endocytosis, so you've got phagocytosis, this one's pinocytosis, and this one's receptor-mediated endocytosis. So they are the three types of endocytosis. What is endocytosis? It's materials that move into a cell in a vesicle. Now remember what a vesicle is? Vesicle is a little bubble, fluid-filled bubble, just like a blister on the back of your heel. That's a little vesicle. And that vesicle contains the material that you want to bring into the cell. So materials move into the cell in a vesicle formed from the plasma membrane. It requires energy supplied by ATP, just the same as active transport. So you could say that vesicular transport is a type of active transport. Now we can have a look here at the diagram, and these are the three types. You'll notice they all bring things into the cell. So this area out here is the outside the cell, this is inside the cell. All of them bring things into the cell, but they do it in a different way, and we'll run through that in the next few slides. Now, exocytosis moves things out of the cell by fusion with the plasma membrane of vesicles formed inside the cell. So a vesicle is formed inside the cell with the particles that it would like to get rid of, that it would like to remove from the cell, and then it gets them out of the cell. You can see here you've got your Golgi apparatus. If you remember, what did the Golgi apparatus do? It's an organelle in the cell. It modified, sorted, and packaged proteins. So if you've got some proteins that are unwanted and need to get them out of a cell, that process is called exocytosis. Golgi apparatus will produce these proteins, or not produce them, but modify them and sort them, put them into a vesicle, and then that vesicle will leave the cell. And the last one is transcytosis. So as I said before, endocytosis on one side and exocytosis on the other. We'll do that a bit more further down the slides. So let's first of all look at receptor-mediated endocytosis. Just bear with me while I bring up my pen and I'll put it into purple. Okay, so let's start at number one. So number one is called binding. Okay, so here we are here. So we've got binding. So what happens here? You've got a ligand. Now, do you remember where you heard the word ligand before? If you go back or have a think back to when you did the functions of proteins, when you did the function of receptor, a ligand attaches to a receptor. So a ligand is any particle or any molecule that attaches to a receptor. In this case here, the ligand is LDL, which is low density lipoprotein. So it goes and attaches to the receptor that exactly matches it. So that receptor will only accept a particular ligand. So it goes and attaches to the receptor and it creates a receptor complex. Okay, so a ligand receptor complex. So at 1, we are left with, you can see here, this is this area here, we have a receptor, and sitting on top of that is the LDL. So you can see here, here are this section here are your receptors and your ligand. Now in number 2, which is over here, what happens here? You can see that the ligand receptor complexes are sitting in what's called a clathrin-coated pit. So what happens in number 2, the edges of the membrane, so you can see here, the edges of the membrane here, okay, they fuse and a small piece of the membrane pinches off and it results in a coated vesicle. So you have the inside here, you can see here, you have the ligand receptor complex, and then on the outside here is the clathrin-coated or clathrin coating. So you've got a vesicle, inside is the ligand receptor complex, and on the outside is the clathrin. What happens then in number 3? The vesicle becomes uncoated. So uncoating, so what happens here? It loses its clathrin coat, hard to say, and that clathrin is then recycled. Okay. So now you're left with a vesicle, an uncoated vesicle with the ligand receptor complex inside. In number 4, that vesicle then fuses with a particle or a structure called an endosome. And what happens in that endosome is, you can see here, it separates. So the actual ligand, which is here, the LDL, separates from the receptor. Okay? So that's what's happening in number 4, and it happens within the endosome because that vesicle has fused with the endosome. Then with number 5, there's recycling of the receptors. So you can see here, these receptors here get recycled into a transport vesicle, and they get put back into the plasma membrane to be recycled into the plasma membrane to be reused. Some of them, okay, go into a transport vesicle here. So you can see here, sorry, the actual LDL, this is the LDL, goes into a transport vesicle. Okay, and that transport vesicle goes to a lysosome. So the vesicle that contains the ligand, it buds off and it fuses with a lysosome. Now, if you remember, lysosome was one of the structures that we learnt about in the cell, and what does it do? If you remember its function, it digests things. So it gets rid of unwanted products in the cell. So what it contains is digestive enzymes, and it breaks down, okay, the molecules. They're broken down and then they're recycled. Okay? So that's receptor-mediated endocytosis. So the next one is called phagocytosis. Now, it is a process in which a cell, and that particular cell is called a phagocyte. Okay? So I'll show you here. Here. So a phagocyte, that's the name of the cell. So phago, if you want to split it up, phago means to eat, cyte is a cell. So it is a cell that eats. So what happens there is that the phagocyte engulfs large solid particles that are on the outside. They digest it and then it expels the waste products. So it happens in 3 steps. So you've got step 1. So step 1 here is in the pink. So this here, you can see, is step 1. Okay, so that's step 1. So in step 1, you've got these things called pseudopods. So the plasma membrane puts out these structures called pseudopods, and it encases and goes around whatever it needs to bring in. Okay, so that could be a microbe, it could be the microbe on the receptor, and it brings it into the cell, and it results in the formation of a phagosome. So that's step 1. Step 2, so you can see step 2 here. Okay, so this is step 2. So step 2, fusion of the lysosome. So a lysosome that's sitting in the cell. So this is the lysosome here. So if you can see here, here's the lysosome. So fusion of the lysosome with the phagosome. So the phagosome's got the unwanted particle in it, and it fuses with a lysosome. Now we know lysosomes contain digestive enzymes. So as soon as that lysosome fuses with it, it goes to step 3. So this is step 3. So the digestive enzymes that are in the lysosome will digest and break down the microbe that was brought in. And then that will leave a residual body, and then that gets removed from the cell. So that is phagocytosis. Now, the particular type of phagocyte that we're talking about, or one type of them, is called a macrophage. And we will learn about them when we do tissues and other systems. Okay? But the particular type of phagocyte that performs this is a macrophage. So they are the 3 steps in phagocytosis. So that's how we get rid of and how we destroy bacteria, things like that. Now, the next type of vesicular transport is called bulk phase endocytosis, and another word for it is pinocytosis. Okay, so the important part that you have to know here is instead of bringing in a particle, it brings in droplets of extracellular fluid. Extracellular fluid being the fluid that is on the outside of the cell. So an example of water. Okay, so it might be bringing in some fluid, and that's why it's called pinocytosis. The way that I remember it, I remember pina colada. You drink a pina colada. Okay, so pinocytosis is drinking. The pino particularly means drinking. It also goes into 3 steps. So in step 1, you can see that the plasma membrane folds inwards, so that's this part here, and it engulfs the extracellular fluid, and what also is dissolved in the extracellular fluid as well. It comes in as well. It forms a vesicle containing a droplet of the extracellular fluid, okay, and also the dissolved solutes. So if you remember what a solute was from the last session, solute is particles. Okay, now, so that's step 1. So this is step 1 here. Okay, that's step 1. Then to step 2, the vesicle fuses once again with the lysosome. Okay, so that's there is step 2. Remembering the lysosome contains digestive enzymes. Okay, and then step 3, the enzymes that are within it. So this is step 3. The enzymes that are in the lysosome then degrade the solutes that are in that little vesicle, and that then exit the lysosome to be recycled. Okay, so that's bulk phase endocytosis, or otherwise known as pinocytosis. So that's your 3 types of endocytosis. So now we'll move on to exocytosis. Remembering exo, external, going to the external. So it is a process in which membrane enclosed vesicles, that's why it's called vesicular transport, it always involves a vesicle. And in this case, it's called a secretory vesicle. So if you have a look at this word here, secretory, because it is secreting it from the cell. It is getting it out of the cell. So what happens, it's the process by which membrane enclosed vesicles form inside the cell. They fuse with the plasma membrane, and then they release the contents into the extracellular. And this is the extracellular fluid here. Okay. So, example of some substances or particles that occurs is neurotransmitters and hormones, okay, that need to be released out of the cell. Now, there are 2 types. So you can see the first one is constitutive, and this one here is constitutive, and then your second one is regulated. Now, the difference between the 2, in constitutive, it occurs when the molecule secreted is needed in constant levels. So to maintain homeostasis, there's that word again, so to maintain balance and equilibrium, that molecule is required all the time. Okay? So examples are albumin. So albumin is a protein that is required for your antibodies. So we constantly need albumin. So that's called constitutive secretion. The other type of exocytosis is regulated. And this is where the protein is secreted in response to a stimulus. So only when it's required is it secreted. So for example here is calcium. Okay, so when calcium's required, only when it's required for whatever reason, it needs more calcium in the blood or something like that, then it will release it. But it's not doing it all the time. It's very regulated. Okay? So you can see here in your constitutive secretion here, you've got the Golgi complex, and here is your proteins. So here is your particles that you want to get rid of. So they are wrapped up into a vesicle. So this is your vesicle here. Okay? So then that vesicle goes and fuses with the plasma membrane, and it releases the product into the extracellular fluid. And it's doing that all the time. Now, regulated, you can see here, there is a signal. So you've got a signal here. So this signal comes and attaches here. Okay? And that signal is what makes the vesicle be created. Okay? So by that signal going and attaching, it tells the cell, okay, I need more calcium. So what happens is, for example, this is the calcium here. The calcium is then bundled up into a vesicle. Then you can see that vesicle then moves to the plasma membrane, and it releases the calcium out into the extracellular fluid. This is happening all the time. It doesn't need a signal. So that's constitutive, whereas regulated needs that signal. An example of regulated exocytosis is with glucose transporters. Remember we did the glucose transporter when we did, if you remember, what type of transport? It was passive, and it was facilitated carrier. So what happens there, the glucose transporter is not required. So here are your glucose transporters here. So these are your glucose transporters, okay? Now they are not required until there is glucose in the blood. So what happens, you've got a heap of glucose, so you've got heaps of glucose all in here, lots and lots of glucose out in the blood. So this is the cell here, and this is the blood. So there's heaps of glucose out here in the blood. What happens is, with glucose in the blood, if there is an increase in glucose, it stimulates the release of a substance called insulin. And that insulin, which is just here, it goes and attaches to an insulin receptor. Okay? Now, here you can see in number 1, let's go down to number 1, the glucose transporters are just waiting in the cell, and they are stored within vesicles in the cell, cell itself. Okay? So they're stored in membrane vesicles. When the insulin interacts, or we're now up to here to number 2, when the insulin interacts with the insulin receptor, the vesicle, so you can see this vesicle here, it moves to the surface, so it goes to the plasma membrane surface, and it puts those, here's your glucose transporter, it puts that glucose transporter into the cell membrane, which means that the glucose transporter is there ready for the glucose, because this is the glucose here, to attach to the GLUT transporter. So when the, okay, so you can see here you've got your glucose attached to your transporter. And then what happens is the glucose then enters the cell. So the glucose goes in, which is where we want it. When the insulin level drops, which means that the blood glucose level in the extracellular fluid, so when the blood level drops of the glucose, insulin level then drops, because you don't need as much insulin then, because you don't have as much glucose out here in the extracellular fluid. So what happens then, the glucose transporters are removed from the plasma membrane, and that's what's happening here. So you can see they're removed from the plasma membrane, and they go back into a vesicle. So by endocytosis, okay, they then come back into the cell. So the smaller vesicles fuse with a large endosome, this is number 4 here, and then they just stay in this form, okay, until required. So patches of the endosome enriched with glucose transporters bud off to become small vesicles, ready for return to the surface when they're required. So that's why it's regulated. So with regulation, it needs a stimulus. So this here is the important part, is you must have a stimulus. And in the case of glucose, it is increased glucose in the blood. Now the last one is transcytosis. So this is where macromolecules, so macro meaning large, molecules being a molecule, contained in vesicles enter the cell via endocytosis, then they move straight across the cell, and then they are released on the other side by exocytosis. Okay? So it occurs most commonly across endothelial cells that line blood vessels. Now one of the main areas this happens is in the placenta. So in pregnancy, in the uterine wall, you have blood vessels. So this here, okay, this is the maternal blood. So this here is your maternal blood, and this is the uterus. So this is the lining of the uterus, okay? And what's attached to that uterus is a placenta. So during pregnancy, antibodies, or IgG, so this is your particle here, okay, they come in from the mother's blood. So what happens is they come in from the mother's blood, okay, into the placenta, they cross the placenta, and then they go into the fetal circulation. Because in pregnancy, the baby's blood does not come in contact with the mother's blood. The placenta is what divides it. So what happens is it goes across the cell, okay, in the uterus, through the placenta, which the placenta is part of that uterus, and then it goes across to the fetal circulation. Okay, some other areas that happen where it happens as well is delivery of IgA, so they're antibodies, to the mucosal surfaces of the respiratory, gastrointestinal and urogenital tracts, and also export into the milk. So it's how your antibodies get into the milk. Okay, so that's transcytosis. If you would like to see a video, at the moment on the screen you've only just got the picture of it, but there's actually a great video if you want to watch it, and it's called The Inner Life of the Cell, and it's got the link here to the YouTube video. So if you have a look here, you can have a look there, and it will show you exactly a representation of what's happening in a cell and how it's constant and it's going on all the time. Okay, so that's it for the content for this session. It's not a very big session. Now what you can do is you can go to your tutorial and you can complete the table in activity 1 and just complete that. And as I've told you before, make sure you keep all those tutorials separate, because that will help you to study for your online quiz. Okay, so preparation for next session, watch the video animation of cell signaling, so you can see activity 1 and session 4, and also review, go back over and review what is a receptor. Because when we're talking about cell signaling, okay, you need to know about the receptors. Okay, so see you next time. Bye.